Hello friends, today we are going to discuss one of the important sub-theme in system physiology, mechanism of urine formation. Mechanism of urine formation. We are all terrestrial animals. Being a terrestrial animal, the chief adaptation is to conserve water. For that, excretion of concentrated urine is a dire requirement. And for that, efficient and functional nephron is must and should or become indispensable. Just like neuron for a brain, nephrons, which are the structural and functional components of kidney. And how these nephrons perform, that means they excrete concentrated urine, we're going to discuss today, and by point, and it will play an important role, as I said, and is a central theme of terrestrial animals adaptation. Basically, mechanism of urine formation involves two steps. One, glomerular filtration. Number two, selective reabsorption. And secretion. We're not going to the basics of nephron structure. We knew that nephron consists of two parts. One, the glomerular capsule and the renal tubule. The glomerular capsule exact means Malpighian body. That means Bowman's capsule plus glomerulus. Whereas renal tubule consists of different parts like proximal convoluted tubule, loop of Henle, that means descending limb, ascending limb, DCT, desiccant tubule, collecting duct. Okay, it's a basic. Okay, but as we go by the discussion, we'll discuss about that. Since our motto is to discuss about urine formation, we'll concentrate on that. And the first phase is, that means how to excrete a concentrated urine is most important. And the first phase is mechanism is through glomerular filtration. Say this is the Bowman's capsule. This is PCT, that means proximal convoluted tubule. And this is loop of Henle. This is descending limb and the ascending limb. This is DCT, that means distal convoluted tubule, and it is collecting duct. This efferent arteriole and the glomerulus is efferent arteriole. So this is Bowman's capsule. This efferent arteriole. Glomerulus. This is PCT, proximal convoluted tubule. This is DCT, distal convoluted tubule. This is descending limb. Ascending limb. Together called loop of Henle. This is collecting duct. These are the different parts of a nephron. This is phase 1 and this is phase 2. There are 1 million nephrons nearly in each kidney. And how they perform? The first phase is the glomerular filtration. Glomerulus, what exactly is called? Tough capillaries formed by efferent arteriole. And this, when the blood passes through these glomerulus, it exerts certain hydrostatic pressure. The first phase is called hydrostatic pressure of blood in glomerulus is 60 mmHg. how the urine get filtered, we have discussed step by step. When the blood passes in the glomerulus, it exits a hydrostatic pressure of 60 mmHg, and it get opposed by the colloidal pressure. That means colloidal pressure means nothing but the blood plasma contains some proteins, it exhibit colloidal pressure is of around 32 millimeters of mercury, and the hydrostatic pressure of Bowman's capsule, is 18 millimeters of mercury, and the net filtration pressure become 10 millimeters of mercury. It is called net filtration pressure. You know how this happens? There are three layers: the endothelial cells of glomerulus and basement, together with basement membrane and podocytes of Bowman's capsule. It contains fenestrations. It contains slit pores. When the blood passes through this glomerular net, it exits this kind of pressure, and this subjected to three kinds of filtration membrane. What triangular filtrate membrane that consists of endothelial cells of blood capillaries, that means within the blood, that means glomerulus, and the basement membrane together with basement membrane, and the podocytes. Podocytes are nothing but the inner lining of the Bowman's capsule, which are unique cells which contains an intricate spaces, minute spaces, and they are called as filtration slits. That means the fenestration is present in the endothelial cells plus basement membrane plus the podocytes filtration slits. Together, these three together leads to the exertion of pressure, which is NFP 10 mmHg, the combination of these three, okay, then ultimately producing primary urine or ultrafiltrate. This is very important, and the process is called globular filtration. You must be learning how this has been done. Simply the blood, it has certain hydrostatic pressure opposed by counter forces. During this process it passes through three different layers: endothelial, basement membrane, podocytes. Then the filtration happens. What exactly and how we came to know that the filtration happens? Because the proteins has been filtered. The essential distinction here is that means in primary urine it is almost similar to blood plasma except proteins. Proteins has been filtered, and the product so far comes out is called primary urine. Primary urine or ultrafiltrate. It is the first phase of urine formation. Then from this onwards it is very important, because how to produce concentrated urine. Concentrated means less water and more dissolved solutes. That means unwanted substances, particularly the excretory substances like urea, creatinine, has to be excreted, and such an excreted urine is a must and should, and it shall be done in phases. That means we have to remove the water. This is very important because we are terrestrial animals, and we have to remove the essential nutrients like glucose, amino acids, vitamins, and we have to add the unwanted excretory substances like creatinine. All these what we call selective reabsorption and tubular secretion. We reabsorb our essential substances like water and nutrients, and we secrete the unwanted substance. That is what exactly called selective reabsorption and secretion. There are different parts in it. All these are renal tubule. It will occur in phases. First, the PCT, what we call proximal convoluted tubule. Then the Henle's loop. Then the DCT. Then the collecting duct. Henle's loop of two types: descending limb and the ascending limb. Here, the PCT in combination with descending limb is most important, because 85% of the filtration, 85% of reabsorption of essential components done by these two tissues. How? For example, in PCT, we'll discuss for one by one. Come here, okay? This is PCT. This is called primary urine, and it enters into the PCT. Proximal convolutules contains epithelial cells with brush borders, microvilli. Cuboidal epithelial cells with brush border, which for increasing area of absorption, and these have different properties. Say, as I told you, we have to reabsorb our essential substances, let's say nutrients. Nutrients have been reabsorbed by active transport. Simply I write here for understanding. Nutrients have been reabsorbed by active transport, and sodium also by active transport, water by osmosis. I told you already, we have to secrete certain components, and here secretion is not only because of excreting the unwanted substance, but also to maintain ionic balance, to maintain ionic and pH balance of body fluid. For that, secretion of H plus, K plus and NH3 is important. These are all reabsorption and these are all secretion. This is very important. Therefore, nutrients has been reabsorbed by active transport, then also the sodium by active transport, then also the water by osmosis. Here we add, that means we secrete H plus, K plus, and also we reabsorb HCO3 minus as well. That means we want water reabsorb back by osmosis, we want our glucose back to transport, then the Na plus back to transport, and secrete H plus and K plus, because there should be a balance, what we call osmoregulation. Is it urine formation is one of the head under homeostasis. For what? They have to maintain an optimum balance between the water and the dissolved solutes, and it will take place in different form. Here is you have to maintain ionic balance and pH balance, that means acid base balance. For that we will secrete certain H plus, K plus and NH3 into the filtrate, into the primary filtrate, and reabsorb the essential substances like glucose, amino acids and water out of the substrate, that means out of the filtrate, because we require that. Ultimately leads to production of concentrated urine through higher osmolarity of the medullary interstitium. Higher osmolarity, this is very important point. Higher osmolarity of medullary interstitium. This is very important, and this is a central theme of producing the concentrated urine. We will discuss in the countercurrent mechanism about this, but for now just listen that this is important. That means higher osmolarity of medullary interstitium is essential for production of concentrated urine. Okay, then this has been done PCT, and in PCT, depending on the essentiality, we require glucose, and they are considered three kind of substances. Thresholds, higher threshold substances like glucose, lower threshold substances like urea, a threshold substances like creatinine. They have to get excreted. So what we call necessity of substances, according to their different types. Okay, PCT is over. Next phase is Henle's loop. The first part is descending limb and the ascending limb. In descending limb, here unlike PCT, not too much reabsorption will take place, but this Henle's loop, as I told you, hyperosmolarity. In the PCT is called cortex part, and this is called in outer medulla, and this is inner medulla. Okay, three different phases. Okay, here the primary urine which comes out of the Bowman's capsule is hypotonic to cortical fluid. Then when it comes to the descending limb, not too much reabsorption takes place, but essential for maintaining the higher osmolarity of the medullary interstitium. Meaning medullary means the PCT enters into the medullary part. It is outer medulla and medulla. Somehow it is medullary interstitium, and here osmolarity is maintaining. Is chief significance of descending limb. That means Henle's loop. How? Here the thing is, it is descending loop. Here water reabsorption only takes place. Here in a descending limb, water reabsorption will take place. That means descending limb of the Henle's loop is permeable to water but almost impermeable to electrolytes. And what is the inference? It leads to the production of a concentrated filtrate when it goes down towards the inner medulla, because water has been reabsorbing in the descending limb. This is very important. I told you concentrated urine will take place in phases. This is one of the phases. Okay, then it enters into the ascending limb. It is reverse. In descending limb, the water reabsorption takes place because it is permeable to water, but in ascending limb, it is impermeable to water but permeable to electrolytes. Here, there are two parts, the thin and the thick. In thin part, the sodium reabsorption will take place by passive transport. In thick part, the sodium reabsorption will take place by active transport. So here, in ascending limb, sodium reabsorption, thin part by passive, whereas in the thick part, it is active. This is very important. Here, what is the inference? It leads to production of diluted filtrate when it enters the cortex. See, because here only solutes, the sodium has been reabsorbing, but the water has not been reabsorbing because it is impermeable, both thin and thick part of ascending limb. Then it moves to DCT. I told you 85% of the reabsorption and urine formation it takes place by the PCT and the descending limb. The rest is called, it is selective. It's like a regulatory mechanism. The 85% is called unregulated. What is regulated? Here there is under the control of hormones. You know, antidiuretic hormone or vasopressin. You know, aldosterone. When the renal filtrate enter the DCT, these hormones come into action. You know, H2O will be reabsorbed in DCT because of ADH, antidiuretic hormone. At the same time, Na plus through aldosterones, Na plus. And here also some H plus, K plus will add into the primary filters just to maintain ionic balance. I told you already. There should be a balance between the water and the ions for the essentiality of body fluids. Here water has been reabsorbed by ADH, antidiuretic hormone. At the same time, ions like H plus, K plus have been adding into the DCT filtrate for maintaining ionic balance. It is about DCT. That means H2O reabsorption by ADH, Na plus reabsorption, then K plus, H plus, they will secrete for ionic balance, ionic and pH balance. Then we come to the collecting duct. I told you concentrated urine will take place in phases. In different parts of the nephron, water has been reabsorbing essential nutrients, ions has been adding, allowing the excrete something like creatinine. That is what we call phase-based concentrated urine. You know, ultimately collecting duct. The same component here also. The ADH will reabsorb the water and allowing the concentrated urine to go further. Ultimately, the concentrated urine will come out. So, you know, phase. In the first phase, that means the globular filtration, the first phase of urine, which leads to the generation of primary urine through three filtrate membrane. One, the endothelial cells of the glomerulus, along with the basement membrane and the podocytes, because the former contain the fenestration. Here the podocytes contain the filtration slits, together form the filtration membrane. Through the hydrostatic pressure at the rate of net filtration pressure of 10 mmHg, the primary urine or the ultrafiltrate has been formed. It is called globular filtration. And the same may be subjected to the next compound, which is chief. Here the reabsorption will take place selectively because we want water, we want glucose, and we don't want certain substances like creatinine. And also to maintain ionic balance, we add H plus K plus into the filtrate concerned. So that's ultimately leads to production of concentrated urine, and it is under the direction or to facilitated by the higher osmolarity of medullary interstitial. We'll discuss in the next issue how it is important for concentrated urine. Okay. So ultimately, then the PCT enters. There is active transport sodium. So as sodium has been actively transported, then the water by osmosis, then the glucose, that means essential substance back to transport. Then the K plus H plus have been added. That means secreted. Then it goes to descending limb, which is completely permeable to water. Water has been reabsorbed due to generation of concentrated filtrate when it goes down to the inner medulla. Then ascending limb, the both thin and thick are permeable to ions like Na plus. Passive transport and thick, active transport and thick. Therefore, water has been conserved. That means it has not been permeated because it is impermeable water. It leads to dilution fluid when it enters in the cortex. That means DCT. Then the AD has directed water reabsorption. At the same time, Na plus also reabsorbed by aldosterone. Then the K plus H plus has been added. Then the collecting duct. Again, the AD directed water has been reabsorbed, and during this mechanism, through the hyper or higher osmolarity of middle interstitium, a concentrated urine will come out. So it's about urine formation. I told you what is higher osmolarity. That means we would understand countercurrent mechanism, because higher osmolarity of the middle interstitium fluid is a must and is essential for the production of or for the excretion of a concentrated urine. So we have to learn about countercurrent mechanism for better understanding how it will be done. It's about different phases of urine formation. Countercurrent mechanism is a typical phenomenon. It is like a head. Countercurrent mechanism for concentrated urine. This is important. We told already. This is Bowman's capsule. This is PCT. Then the descending limb. Then the ascending limb. BCT and also. Okay. Just focus on the loop of Henle. This is called loop of Henle. And this is medullary interstitium. This is cortex part. This is descending limb. This is thin and thick ascending limb. This is DCT. Then this is collecting duct. Then we have to discuss about vasa recta. It's a blood vessel, This is nephron and this is vasa recta. Now how it happens? Here the primary urine will flow from descending limb then to the ascending limb. That means counter or opposite direction. Ascending limb then descending limb. Opposite direction now. Similar to that, in vasa recta also the blood flows here. Opposite direction, that means counter. This is the descending part. This is the ascending part. This counter mechanism leads to creation of higher osmolarity of medullary interstitium. For example, in cortex it is 300 osmolarity, but in medulla it is 1200 osmolarity. That means from 300 to 1200 there is a rise. How this happens? This is very much important in understanding for concentrated urine. For example, we told you already This arrangement is important first because the vasa recta and the Henle's loop are close to each other. What if they are close to each other? There is a matter for concern, you know. In thin, here there is a secretion of NaCl. That means in ascending limb, we knew that we have discussed NaCl has been reabsorbed, and this NaCl will enter. That means the sodium will enter into the descending limb of vasa recta. Then it will enter the ascending limb. Then again it come back to the interstitium. That means the sodium which has been reabsorbed in the ascending limb of loop of Henle of nephron will enter the descending limb of vasa recta, then the ascending limb, and get back to the interstitium. That means increase in the osmolarity. Next part, the urea. Same case of urea also, which will enter into the ascending limb of nephron. It will travel, say DCT, and again collecting duct. Again it will get reabsorbed and enter into interstitium. That means Na plus and urea are crux point for maintaining higher osmolarity. Of medullary interstitium. This is very important. That means the solute concentration, that means the concentration gradient, what we call interstitial concentration gradient, is required. Because if this happens, which facilitates the easy reabsorption of water, this is very important. When this, because of NaCl, which will reabsorb in ascending limb and goes down the descending limb of oscillator, in ascending limb it will reabsorb. At the same time, urea has been reabsorbed in the ascending limb, then DCT, then collecting duct, then again reabsorbed in interstitium. All these facilitate easy water reabsorption in the collecting duct. Is very important. Easy water reabsorption. Now, to facilitate the easy water reabsorption, such a counter current, opposite direction, in case of Henle's loop and the vasa recta. In case of Henle's loop, it is called counter current multiplier. Such an arrangement is essential for production of concentrated urine. It's very important. Okay, and we are talking about urine. What exactly urine and how it gets released out? Okay, this is the last part of the urine formation. You know, urine is nothing but 96% of water and 2% of urea and 2% of other substances, and its pH is 6.0. that means slightly acidic and light yellow color. Urine is important in identifying the clinical diagnosis. For example, glucosuria, ketonuria, that means diabetes mellitus, and how the urine get released, what we call micturition process. Micturition. The release of urine is called micturition, facilitated by the stretch receptors present in urinary bladder. This is very important. You know, we feel urinated. It is called, the release of urine is called micturition. How this happens? When the urinary bladder gets filled with urine, it sends a signal to central nervous system. For example, this urinary bladder, then CNS. The central nervous system releases motor signal, a voluntary signal, motor, to the smooth muscles of urinary bladder. How it sends? Because stretch receptors will send to CNS. Because they feel that urinary bladder is full, so it has to get relaxed. Then the smooth muscle get contracted. At the same time, urethral sphincter, what we call urethral sphincter relaxation. Urethral sphincter relaxation. Ultimately, the urine comes out. The whole process is called micturition. It is called micturition reflex. It's a voluntary signal of CNS through the stretch receptor mediation. Okay, so it's all about why we require such a laborious process. Why we require constant urine? Because we are terrestrial animals, and it's called osmoregulation, maintaining of water quantity and the dissolved solutes in balance is osmoregulation. It will be, in case of ourselves, that means through urine it will be done. Apart from it, through sweat it also performed. And in case of urine, how the water has been regulated, how the solutes has been dissolved solutes has been regulated by hormones like ADH. We already discussed antidiuretic hormone. Apart from it, we also discuss aldosterone and angiotensin 2. Angiotensin 2. These are all required for osmoregulation. That means a concentrated urine is very important. So it's about the urine formation. I already told you in the lactase class, follow a standard book, the same time a basic book, so that through my lecture you can consolidate your knowledge. My objective is to serve to poor and talented students who cannot afford the topmost fees or higher fees for topmost CSR exams and other top exams. Okay, you may like, comment. Thanks for watching.